Hello. Immunization for vaccine preventable diseases. This is the topic of discussion today. Immunization protects children from serious and sometimes fatal infectious disease by stimulating the immune system to develop or to produce antibodies and protect them before they are exposed to the diseases. This is a long and also important class, so I'll divide this class into 3 to 4 parts. So I'll take 3 or 4 lectures. Today I'll start with the basics and then definitions and then classification of vaccine. So what is immunization? To define immunization, this is technique by which immunizing agents are introduced into the body for inducing immunity to produce antibody artificially, or preformed antibodies are given to prevent the child from diseases. Why to immunize a child? Number 1 point is it increases level of general immunity. Another, widespread use of vaccines will help you for global eradication of diseases. Examples are smallpox, which has been eradicated from the world, and polio also almost eradicated. And it minimizes the spread of infection. So it is very necessary to immunize a child. Let's discuss the immunity and immune system of the body before going to the vaccination in detail. Immunity is a mechanism of body by which it recognizes an immunogen and tries to eliminate it. Types of immunity: one is natural, innate or the inborn immunity, and another is acquired immunity. The natural or the innate immunity is inborn. So the components of this immunity will be skin, mucous membrane, cilia, phagocytosis, macrophages, neutrophils, natural killer cells, inflammation, normal flora, tears, saliva, cytokines like interleukins and tumor necrosis factor, interferons, chemokines, interleukin 8, and the complement systems. So they are protecting in different ways, and these are all inborn congenitally, that is by birth an individual has. And acquired immunity is again divided into active and passive. Active is antibodies are produced by the body itself, and passive is preformed antibodies are introduced to the body. The difference between active and passive immunity. Active immunity, there is an active participation of the host. Like example, after a vaccination or a natural infection, the host participates to produce antibody. Whereas in the passive immunity, there is no host participation but preformed antibodies are injected. Example is immunoglobulins. An onset of action for active immunity is longer. After introduction of the vaccine, it takes some time for immunity to develop. Whereas in the passive immunity, the action is immediate. As soon as you give the immunoglobulin, the action starts. And duration of immunity is long-lived in case of active immunity. It lasts for months to years. Example is vaccination and also the natural infections. Whereas passive immunity, the duration is short-lived. It is just for weeks. So example is immunoglobulins you give from outside, and sometimes even the immunoglobulin that crosses through the placenta. That is a natural passive immunity. Example of natural passive immunity is the immunoglobulin passing across the placenta. Okay, in short, immunity, innate, congenital, and then acquired. Acquired again active and passive. Active example is vaccine and natural diseases, and passive is the immunoglobulin passing through the across the placenta and also the different immunoglobulins that are prepared. Now come to immune system of the body. These are network of cells which recognizes and shows immune response to introduced immunogens. The cells of the innate immune response are macrophages, dendritic cell, NK cell, that is natural killer cell, mast cell, basophil, eosinophils. Cells of acquired immune responses are lymphocytes. A, B cell, that is converted to plasma cells, and then they secrete the different immunoglobulins like IgM, IgG, IgA, IgD, IgE. B, T cell. So T cell can again be of helper T cells, that is CD4, cytotoxic T cells, CD8, and regulatory T cells, previously known as the suppressor T cells. So they suppress excess immune response. And then C, memory B and T cells are developed. So these are all classification of the lymphocytes or the cells of acquired immune response. Immune response can be primary response and secondary response. It's very important to understand this because the vaccine works in this way. So primary response is when an individual is exposed to an antigen for the first time. It can be first dose of the vaccine or any infection or any viral infection, bacterial infection. So exposed for the first time, then there is a lag period of 5 to 7 days before the antibody is produced. So after this lag period, antibodies are produced, and in primary response the type of antibodies produced are of IgM type, and the duration of action is lasting. These are transient in action, so the duration of immunity is short-lived. So they last for few days, 2 weeks, and then they wane away. Then comes the secondary response. So same antigen reintroduced again. Suppose second dose of vaccine is given or person is infected by the same virus again. So that time the lag phase will be shorter, and then antibodies are produced, and the type of antibodies produced are IgG type, and this persists in the serum for months to years. So the immunity will be long lasting in secondary response. You will better understand if you see this graph. So this graph is showing you the primary and secondary response. So here, first dose of vaccination or any infection, there is a lag period, and then gradually the immunoglobulins are formed, that is of IgM type in the primary response. Then this antibody is short-lived, so it wanes away. And then at this point, if you give the second dose of vaccine, or secondarily the child is infected, then this time the lag phase is very short. Suddenly you get a high amount of antibody, and then rapidly it rises, and the type of antibody you get is IgG type. So the function of this is long lasting, so this persists for longer time. So this, when you give second, third dose of vaccines, these things are happening. So you have to understand what is primary and what is secondary response. Now the immunizing agents. Immunizing agents are classified as active immunization, passive immunization. Example of active immunization are the vaccines, and the example of passive immunization is immunoglobulins and antiserum or antisera. So if you see here, immunizing agents, vaccines are the active immunizing agents, and passive are immunoglobulins and antisera. So all these are coming under immunizing agents. So slowly I'll discuss vaccines first, then followed by immunoglobulins and antisera. Vaccine is more important. Let's start with vaccines. These are biological preparation of attenuated, live, or killed inactivated microorganisms or antigenic portions of these agents presented to a potential host to induce immunity and prevent disease. So this is definition given by WHO. A vaccine typically contains an agent that resembles a disease-causing microorganism. The beginning of the vaccine. Year 1796. This is the year of the beginning of vaccine, when Edward Jenner who is considered as founder of vaccinology. He first inoculated a 13-year-old boy with vaccinia virus, that is cowpox virus, and then demonstrated immunity to smallpox. So this was the beginning of the vaccination. And in year 1798, the first invented vaccine was against smallpox. So that developed in 1798. You can see in the picture how bad the smallpox rash is. So they used to have lots of mortality because of smallpox. So this was the first invented vaccine in 1798. Then the scientific era for vaccination started when Pasteur demonstrated that microorganisms cause disease, and then he discovered and made vaccines weakened or attenuated, and then the earliest vaccines developed were against cholera, anthrax and rabies. Let's start with the active immunization. So active immunization examples are vaccination and then the infections. Infections are the natural active immunity. Here the immunity is acquired after recovery from natural infections like viral, bacterial, fungal, parasitic, any infections you'll acquire immunity. Whereas vaccination, it is a process of administering a vaccine or the immunogens to stimulate immune system to produce antibodies and cellular immune responses. That means humoral as well as the cellular immune responses are produced against a specific disease. So that process is known as vaccination. Whereas passive immunity is injection of the preformed antibodies in the form of antiserum or immunoglobulin to provide immunity to the body. And the example of natural passive immunity is the transplacental transmission of antibodies from mother to the fetus, which again provides protection to the baby for first 3 to 6 months of life. So this is example of passive immunization. So if you see the graph, active and passive immunization, so this blue line, this is a passive immunization. So soon after you give, there is a high antibodies, so which is going to wane up very early. So this is very short-lived. Whereas in case of active immunization, initial injection with the primary response, secondary response with booster doses, you get a higher amount of antibodies, and then that is persisting for longer duration. Now come to the classification of vaccines. This is very important for exams and also for life. So number 1, live attenuated vaccines. Number 2, killed and inactivated vaccines. Number 3, you get subunit vaccines. Example of subunit vaccines are toxoid vaccines, recombinant vaccines, polysaccharide vaccines, conjugate vaccines and other subunit vaccines. Number 4 is combination vaccines. Number 5, nucleic acid vaccines. So again now I'll discuss in detail about all these vaccines. Let's start with the live attenuated vaccine. These vaccines are created by reducing the virulence of pathogen, that is attenuation, but it keeps the pathogen alive. They are still viable, but they are less virulent. So live vaccines contain weakened or attenuated but living organism that can replicate in the body of the individual but are harmless or less virulent, so it does not cause the disease. The mechanism of action of live vaccines are: first, they replicate slowly in the host but does not cause disease. Then these antigens of the vaccine is taken up by antigen presenting cells, which are macrophages and dendritic cells. After that, there is activation of immune system. Both B and T cell activation occurs, producing humoral as well as cell mediated and also sometimes local immunity. Examples are intestinal mucosa by the oral polio vaccine. Then memory cell formation takes place. Both B and T cell memory cells are formed. Then there is a long-lasting protection. So on exposure to a real pathogen, you get a immune system responding. which responds rapidly and strongly, and then protects the child from any disease. Immunity is achieved with 1 or 2 doses. So in case of live vaccines, the duration of action is longer and it is strong, so just 1 or 2 doses is sufficient. Many times maybe just 1 dose is also sufficient. The advantages of live vaccine is it induces a broad immune response, both cellular and humoral. Low dose is required. Low dose is sufficient. And then it provides strong and long-lasting immunity. Disadvantage is it can cause sometimes adverse effects, especially in the immunocompromised people. So it is not given, it cannot be given to the immunocompromised individuals. Classification of live vaccines. It can be bacterial, it can be viral. So bacterial example is BCG vaccine, TY21A typhoid vaccine, this is the oral typhoid vaccine, and plague vaccine. Viral live vaccines, live attenuated vaccines are oral polio vaccine, MMR, that is measles, mumps, rubella, rotavirus, varicella zoster, yellow fever, and then Japanese encephalitis with the strain SA14-14-2. So this one is the live vaccine, and this one we use in the immunization schedule. Now killed vaccines. Killed vaccine contain dead pathogens. The virus particles or the bacteria or other pathogens are grown in culture and then they are killed or inactivated by heat or formaldehyde, but they remain antigenic. The special features are they are safer than the live vaccines, but they usually need booster doses. Classification of killed vaccines are bacterial killed vaccines are Bordetella pertussis, Salmonella typhi, and Vibrio cholera. And then viral vaccines which are killed are Japanese encephalitis, injectable poliovirus, rabies, and hepatitis A. Let's see the difference between the live and the killed vaccines. Number and quantity of dose. In case of live vaccine is usually single and low dose is sufficient. But killed vaccines requires multiple doses and then high doses, higher doses than the live vaccine. Antibody persistence or the duration of immunity in live vaccine is longer, killed it is shorter. Booster needed in case of killed vaccines, in case of live very infrequently. Cell-mediated and mucosal immunity production is there in the live vaccines, not there in the killed vaccines. Excretion of vaccine virus and transmission to non-immune contacts are possible in case of live vaccine and no in the killed vaccine. Immunoglobulins produced are IgA, IgG type in case of live vaccines, and it is IgG type in killed vaccine. Stability at room temperature is low in case of live vaccine and then high in case of killed vaccine. Usually live vaccines are freeze dried, it is kept in the colder compartment. Need for adjuvant? None in case of live vaccine. In case of killed vaccine, yes, because the adjuvant is going to enhance the immunity. Third type of vaccine was subunit vaccines. So there are different subunit vaccines. Subunit vaccines, they use specific antigens, either protein or polysaccharide of the pathogen. So example number 1, toxoid vaccines. Here the toxins are inactivated. They are prepared by detoxifying the exotoxin of the bacteria, making them antigenic but not pathogenic. And always adjuvant is used to increase the potency of the vaccine. And the antibodies which are produced here, it neutralizes the toxins, but it does not act on the organism. It does not kill the bacteria, but they neutralize the toxin. So toxoid based vaccine example is vaccine against diphtheria and then tetanus toxoid, Clostridium tetani. So tetanus and diphtheria vaccines are toxoid based vaccines. Number 2, surface antigen or recombinant vaccines. These vaccines are produced using genetic engineering. It is prepared by cloning the antigen. Example, hepatitis B, HBsAg antigen gene in the yeast cell. So this HBsAg which are produced is then used for vaccination or the vaccine preparations. The example of recombinant vaccines are hepatitis B vaccine and human papilloma virus vaccine. Number 3, polysaccharide or polypeptide cellular fraction vaccines. These contain the polysaccharide capsule of bacteria, extracted cellular fractions. Examples, meningococcal vaccine contains the polysaccharide antigen of the cell wall. Similarly, pneumococcal vaccine contains the polysaccharide in capsule of bacteria. So the example of these vaccines are pneumococcal polysaccharide vaccine, that is the PPSV23. Then meningococcal polysaccharide vaccine, that will be against ACY.W135 strain. And then typhoid VI polysaccharide vaccine, and then Haemophilus influenza type B vaccine. The special features are these are poorly effective in case of children less than 2 years, so cannot be used in the immunization schedule. So these are optional vaccines which are given later. And then immunity is poorer than the conjugate vaccine in this polysaccharide vaccines. Next is the conjugate vaccines. Conjugate vaccine contain the polysaccharide antigen but linked to a protein carrier. Specific protein carrier is used to link this polysaccharide, so they are more stronger. Example, Hib conjugate vaccine, pneumococcal conjugate vaccine, meningococcal conjugate vaccine. The special feature of these conjugate vaccines are immunity is strong, long lasting, and this is effective in less than 2-year-old children. So these vaccines are used in the immunization program. So the Hib vaccine, pneumococcal vaccines which are present in the immunizing schedule are these conjugate vaccines. Some other subunit vaccine are acellular pertussis vaccine and shingle vaccines. Now come to combination vaccines. Especially this is designed for the developing countries. More than one kind of immunizing agents is included in the vaccine. Examples are pentavalent vaccine which is used in the immunization schedule, DPT, DT, PD, these are other combinations, MMR or MR, so these are other combinations. The special features are it protects against more than one disease in a single injection. So it simplifies the administration. So one injection you get protection against so many diseases. So it is easy for the child as well as the parents also, because it requires less visits. It is cost effective also, reduce the cost. And child requires just one prick, so and then even for the parents it is helpful. 5. Nucleic acid vaccines. It contains genetic material that code for an antigen. Example, messenger RNA COVID-19 vaccines. Contraindication of live vaccines. Number 1, persons who are immunocompromised, who has immunodeficiency diseases. Persons where immune response is depressed, like in malignancy, corticosteroid therapy, radiation or severe malnutrition, and the pregnant ladies. Kill vaccine, actually there are no contraindication of kill vaccines, but if there is a severe local or general reaction to the previous dose, then it is contraindicated. Let's see classification as per method of preparation. So based on the method of preparation, again vaccine can be classified as a whole cell vaccine. Number 2, split vaccine. Number 3, subunit vaccine. I have already discussed this, so I will not discuss this in detail. And some other type of preparations. Whole cell vaccine, let's start with the viral whole cell vaccines. So in a whole cell vaccine, the vaccine contains entire virus. can be live attenuated or killed or inactivated. So live attenuated whole cell viral vaccines are MMR, OPV, oral polio vaccines, varicella vaccine, rotavirus, and these viruses can replicate in the body and they also have a strong immunity. They replicate in the body but do not cause disease, and these have a strong immunity. Inactivated killed whole cell vaccines are injectable polio vaccine, rabies, hepatitis A. They cannot replicate and they usually need multiple doses. Viral split vaccines, made by breaking virus into pieces. Usually envelope or protein parts are used, and these cannot replicate. So example of this is split influenza vaccine, and this needs booster doses. And subunit viral vaccine I have already discussed, contain only specific purified component of the pathogen and the protein antigen. This cannot replicate. An example was hepatitis B virus and human papilloma virus vaccine. Now come to bacterial whole cell vaccine. They contain the entire bacteria, either live, attenuated or inactivated killed. They have strong immunity but they can show more side effects. So whole cell killed vaccines are cholera and pertussis vaccine. Whole cell live attenuated is BCG vaccine, oral polio vaccine. Oral typhoid vaccines. Whole cell live attenuated vaccines are BCG and oral typhoid vaccines, TY21A. The split fractional bacterial vaccines, these are made by breaking bacteria into pieces. Usually envelope or protein part is used. These are inactivated. It is safer than whole cell vaccines, but these are very rarely used. And subunit bacterial vaccines I have already discussed in detail above. And these are subunit vaccines are usually used in the scheduled vaccination for children. And some other vaccines based on the preparation method are polyvalent vaccines. These are prepared from 2 or more strains of same species. Example, polio, influenza vaccine, pneumococcal vaccine. Others are autogenous vaccine, when organism is obtained from the same person. This is known as autogenous vaccine. And adjuvant or adsorbed vaccines. Here adjuvants are added. So these are substances, when these are added, it enhances the immune response of the vaccine. So the adjuvants used are aluminum phosphate, aluminum hydroxide or alum. The example of this type of vaccine is DT, hepatitis B and acellular pertussis. Other is freeze-dried vaccines. They are more stable preparation than the liquid vaccines. Example of freeze-dried vaccines are BCG, yellow fever, measles vaccine. Okay, thank you. So next class I'll be discussing on the cold chain and the vaccine vial monitors, and then maybe the route of vaccination, schedule of vaccination. Thank you.